Assalamualaikum friends, welcome back to my YouTube channel. Friends, hope so you are all doing well. In the previous video we are discussing about the proximal convoluted tubule, its structure and its function like reabsorption. This is my next video, and in this video we are discussing the further functions like co-transport. Now we will move toward the co-transport. Co-transport refers to the movement of multiple solutes through the same channel. There are two types of co-transporters: symporters and antiporters. Symporters that move two or more molecules in the same direction, for example SGLTs, and antiporters transport that move two or more molecules in opposite direction. Symporters in same direction and antiporters in opposite direction. The sodium concentration gradient allows other molecules such as glucose to be transported across the apical membrane against their concentration gradient. For example, SGLT transporters move glucose together with two sodium ions across the apical membrane. Glucose then crosses the basolateral membrane via facilitated diffusion. Sodium and amino acid symporters are present on the apical side of cells in the S1 segment of the PCT, which reabsorbs all the amino acids in the PCT. Sodium and hydrogen antiporter is found on the apical surface of the PCT cell. It is an antiporter and therefore transport ions across the cell membrane in the opposite direction. In this case, the N-positive ions move into the tubular cells while the H-positive is expelled into the lumen. The primary function of this transporter is to maintain the pH. This is the co-transporter function of the proximal convoluted tubule, and next function is the movement of water. What is the movement of water? Movement of water in the PCT, large volumes of solute are transported into the bloodstream. This means that as we move along the tubule, solute concentration in the tubule decreases, and while the solute concentration in the interstitium increases. The difference in concentration gradient results in the water moving into the interstitium by osmosis. Water mainly takes the paracellular route to move out of the renal tubule, but it can also take the transcellular route. You can see clearly in the next diagram that we are discussing. First of all, sodium enters from the left side, and it can be move out from the cell, and one ATP molecule enter with the potassium. Next you can see that bicarbonate and hydrogen expel out. You can see the diagram showing ion absorption and secretion with the proximal convoluted tubule in the diagram. Next we are discussing about the secretion. Secretion is when substances are moved from the blood and wire transported into the PCT. This is very useful as only 20% of the blood is filtered in the glomerulus every minute. So this provides an alternative route for the substances to enter the lumen. The PCT secretes organic acid, hydrogen ions, and drugs and toxins. First of all, organic acid and bases: bile salts, oxalate, and catecholamines. Hydrogen ions important in maintaining acid base balance in the body. Hydrogen secretion allows reabsorption of bicarbonate via the use of the enzyme carbonic anhydrase. The net result is for every one molecule of H positive secretin and one molecule of bicarbonate, and Na positive is reabsorbed into the bloodstream. As the H positive is consumed in the reaction in the tubular lumen, there is no net excretion of H positive. In this way, about 85% of filtered bicarbonate is reabsorbed in the PCT. Drugs and toxins: secretion of organic cations such as dopamine or morphine occurs via the H positive OC positive exchanger on the apical side of the tubule cell, which is driven by the sodium and hydrogen antibutyl. This is all about the functions of the proximal convoluted tubule. Inshallah, we will meet in the next video. In the end, I request to my lovely friends to subscribe my channel, like my video. Thank you so much.